Dear learners, in this video we are going to study chapter 10 biotechnology and its applications. So in this chapter we will cover some four topics that are technological applications in agriculture, second is biotechnological applications in medicine, and transgenic animals ethical issue. So here we start our chapter. Biotechnology, as you would have learned from the previous chapter, essentially deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. The applications of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremedication, waste treatment, and energy production. Three critical research areas of biotechnology are: first is providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Second is creating optimal conditions through engineering for a catalyst to act. And third is downstream processing technologies to purify the protein or organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially the field of food production and health. Biotechnological applications in agriculture. Let us take a look at three options that can be thought for increasing food production. First is agrochemical based in agriculture, second is organic agriculture, and third is genetically engineered crop based agriculture. The Green Revolution. The The Green Revolution succeeded in tripling the food supply, but yet it would not thrive to feed the growing human population. Increased yields have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals, fertilizers, and pesticides, basically. However, for farmers in the developing world, agrochemicals are often too expensive and further increases in yield with existing varieties are not possible using conventional breeding. As traditional breeding techniques fail to keep pace with demand and to provide sufficiently fast and efficient system for crop improvement. Another technology called tissue culture got developed. What does tissue culture mean? It was learned by scientists during 1950s that whole plants could be regenerated from ex plants. It means any part of a plant taken out and grown in a test tube under sterile conditions in special nutrient media. This capacity to generate a whole plant from any cell or explant is called totipotency. You will learn how to accomplish this in higher classes. It is important to stress here that nutrient medium must provide a carbon source such as sucrose and also inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. By application of these methods, it is possible to achieve propagation of large number of plants in very short duration. This method of producing thousands of plants through tissue culture is called micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown. It means they are somaclones. Somaclones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Try to visit a tissue culture laboratory with your teacher to better understand and appreciate the process. Another important application of the method is the recovery of healthy plants from diseased plants. Even if the plant is infected with a virus, the meristem, apical, and axillary is free of virus. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plants. Scientists have succeeded in culturing meristems of banana, sugarcane, potato, etc. Scientists have even isolated single cell from plants, and after digesting their cell walls have been able to isolate naked protoplast surrounded by plasma membranes. Isolated protoplasts form two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplast, which can be further grown to form a new plant. These hybrids are called somatic hybrids. While the process is called somatic hybridization. Imagine a situation when a protoplast of tomato is fused with that of potato, and then they are grown to form a new hybrid plants combining tomato and potato characteristics. Well, this has been achieved, resulting in the formation of pomato. Unfortunately, this plant did not have any desired combination of characteristic for its commercial utilization. Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield for their fields? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects in the environment are reduced? Use of genetically modified crops is a possible solution. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. GMO. GM plants have been useful in many ways. Genetic modification has first made crops more tolerant to abiotic stresses, cold, drought, salt, and heat. Second, reduced reliance on chemical pesticides, pest resistant crops. Third, help to reduce post harvest losses. Fourth is increase the efficiency of mineral usage by plant. This prevents early exhaustion of fertility of soil. Fifth is enhanced nutritional value of example gold rice. It means vitamin A enriched rice. In addition to these uses, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, foods, and pharmaceuticals. Some of the applications of biotechnology in agriculture that you will study in detail are the production of pest resistant plants, which could decrease the amount of pesticide used. BT toxin is produced by a bacterium called a Bacillus thuringiensis, BT for short. BT toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticides. In effect, created a biopesticide. Examples are BT cotton, BT corn, rice, tomato, potato, and soybean, etc. BT cotton. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as Lepidopterans, tobacco woodworm, armyworm, Coleopterans, beetles, and Dipterans, flies, mosquitoes. B. thuringiensis, B. thuringiensis forms protein crystals during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill bacillus? Actually, Bt toxin protein exists as inactive protoxins, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of gut, which solubilizes the crystals. The activated toxin binds to the surface of mid-gut epithelial cells and create pores that cause cell swelling and lysis and eventually cause death of the insect. Next is specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes depend upon the crop and targeted pest, as most Bt toxins are insect group specific. The toxin is coded by a gene CRY named CRY. There are a number of them. For example, a protein encoded by the genes CRY and CRY first AC and CRY second AB control the cotton bollworms. That of CRY first AB controls corn borer. This is a cotton ball. This is a cotton ball. Oh, sorry. This is a cotton ball A. This this one is destroyed by bollworms, and this one is fully mature cotton ball. Understood? So, coming on our topic, pest resistant plants. Several nematodes parasite a wide variety of plants and animals, including human beings. A nematode, Meloid digne incognitia, infects the root of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference, RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to a complementary dsRNA, the molecule that binds to and prevents translation of mRNA silencing. The source of this complementary RNA could be from an infection by viruses to have RNA genomes or mobile genetic elements, transposons. That replicate via an RNA intermediate. Using agrobacterium vectors, nematode specific genes were introduced into host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double stranded dsRNA that initiated RNA and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Figure 10.2. This is the figure 10.2. A shows the host plant generated DSRN triggers protection against nematode infestation. A is the roots of typical control plants. These are the roots of typical control plants, and B is transgenic plant root 5 days after deliberate infection of nematode but protected through novel mechanism. Okay, biotechnological applications in medicines. The recombinant DNA technological process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant that therapeutics does not induce unwanted immunological responses as is common in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been improved for human use the world over. In India, 12 of these are presently being marketed. Genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time intervals. What would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from other animals be just as effective as that secreted by the human body itself, and would it not elicit an immune response in the human body? Now imagine if bacterium were available that could make human insulin. Suddenly the whole process becomes so simple. You can easily grow a large quantity of bacteria and make as much as insulin as you need. Think about whether insulin can be orally administered to diabetic people or not, and why. Insulin was used for diabetic was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source through caused some patients to develop allergy or other types of reactions to the foreign protein. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges, figure 10.3. This is the figure 10.3. In mammals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also needs to be processed before it will become a fully mature and functional hormone, which contains an extra stretch called the C peptide. This C peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for the production of insulin using rDNA techniques was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. Chains A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. Gene therapy. If a person is born with a hereditary disease, can corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase ADA deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which a functional ADA is given to the patient by injection. But the problem with both of these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of patient are grown in culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolated from marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. Molecular diagnosis. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis, serum and urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction, PCR, and enzyme linked immunosorbent assay ELISA are some of the techniques that serve the purpose of early diagnosis. The presence of pathogen, bacteria, viruses, etc. is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PR. Can you explain how PCR can detect very low amounts of DNA? PCR is now routinely used to detect HIV as suspected AIDS patients. It is being used to detect mutations in genes and suspected cancer patients. 2. It is a powerful technique to identify many other genetic disorders. A single stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear in the photographic film because the probe will not have complementarity with the mutated gene. Analysis is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins, etc., or by detecting the antibody synthesized against the pathogen. Next is the transgenic animals. Animals that have had their DNA manipulated to possess and express extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, fish have been produced. Although over 95% of all existing transgenic animals are mice. Why are these animals being produced? How can man benefit from such modifications? Let us try and explore some of the common reasons. First is normal physiology and development. Second is study of disease. Third is biological products. Fourth is vaccine safety. Fifth is chemical safety testing. And that is finished. Normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affected the normal function of the body and its development. Example study of complex factors involved in growth, such as insulin like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects that result from information is obtained about the biological role of the factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as models for human disease so that the investigation of new treatments for disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Biological products. Medicines required to treat a certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by introduction of portion of DNA or genes which codes for a particular product, such as human protein alpha 1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria PKU and cystic fibrosis. In 1997, the first transgenic cow, Rossi, produced human protein enriched milk, 2.4 grams per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than the natural cow milk. Next is vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of polio vaccine. It's successful and found that reliable that could replace the use of monkeys to test the safety of batches of the vaccine. So this is known as toxicity or safety testing. The procedure is the same as the used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time. Ethical issues. The manipulation of living organisms by the human race cannot go on any further without regulations. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organisms such as GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of gene research and the safety of introduced GM organism for public services. The modification or usage of living organisms for public services as food and medicine sources, for example, has also created problems with patients patents granted for the same. There is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic materials, plants, and other biological resources that have long been identified, developed, and issues used by farmers and indigenous people of a specific region or country. Rice is an imported food grain, the presence of which goes back thousands of years in Asia's agricultural history. There are an estimated 2 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. There is a reference to basmati in ancient text folklore and poetry as it has been grown for centuries. In 1997, an American company got patent rights on basmati rice to the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed with semi-dwarf varieties and claimed as invention or a novelty. The patent extends to functional equivalents, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent uses, products, and processes based on Indian traditional herbal medicines. Example: turmeric, neem. If we are not vigilant and we do not immediately counter these patent applications, other countries or individuals may in cash or may in cash or one second. Uh, in cash on our rich legacy, and we may not be able to do anything about it. Biopiracy is the term used to refer to use of bioresources by multinational companies and other organizations with proper authorization from the countries and people concerned without the compensatory payment. Most of the industrial nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during their commercialization. There has been growing realization or the injustice in adequate compensation and benefit sharing between developed and developing countries. Therefore, some nations are developing laws preventing such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patents Bill that takes issues and consideration, including patent term of emergency provision, research and development initiatives. Coming to the summary, biotechnology has given to humans several useful products by using microbes, plant, animals, and other metabolic machinery techniques and tissue culture and somatic hybridization offer vast potential for manipulation of plants in vitro to produce new varieties. Recombinant DNA technology has made it possible to engineer microbes, plants, and animals such as that they have novel capabilities. Genetically modified organisms have been created by using methods other than natural methods to transfer one more gene from an organism to another, generally using techniques such as recombinant DNA technology. GM plants have been useful in increasing crop yields, reduce post-harvest losses, and make crops more tolerant of stresses. There are several GM crop plants with improved nutritional value of foods, reduced reliance on chemical pesticide, pest-resistant crop. Recombinant DNA technological processes have made immense impact on the area of healthcare by enabling mass production of safe and more effective therapeutics. Since the recombinant therapeutics are identical to the human protein, they do not induce unwanted immunological responses and are free from risk from of animal, as was observed in case of similar products isolated from non-human sources. Human insulin is made up in bacteria, yet its structure is absolutely identical to that of the natural molecule. Transgenic animals are also used to understand how genes contribute to the development of a disease by serving as models for human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Gene therapy is the insertion of genes into an individual cell and tissues to treat diseases, especially hereditary diseases. This does so by replacing a defective mutant allele with a functional one or gene targeting, which involves gene amplification. Viruses that attract their host and introduce their genetic material into the host cell as part of their replication cycle are used as vectors to transfer healthy genes or more recently portions of genes. The current interest in the manipulation of microplant and animals has raised serious ethical questions. Our chapter